Hello everyone and welcome. In this video we're checking out variant 3 of October-November 2024. Before starting, make sure to check out the free notes I have available for you in the comment section down below. Now let's go ahead and start with question number 1. Which steps are needed to find the actual width of a xylem vessel viewed in a transverse section using a times 10 objective lens? Okay, convert from millimeter to micrometer by multiplying by 10 to the negative 3. Actually, you should multiply by 10 to the power of 3, not 10 to the negative 3, because 10 to the negative 3 is like dividing by 1,000. So this is wrong. Calibrate the eyepiece graticle using a stage micrometer on a times 4 objective lens. Now he's just stated that he said it is using a times 10 objective lens. So why would you change the magnification that you're using? No. Measure the width of the xylem vessel using an eyepiece graticle. That is right, you should measure the width using an eyepiece graticle and then multiply the number of eyepiece graticle units by the calibration of the eyepiece graticle, so it's 3 and 4 only. Now, which cell structure is found in human cells and a typical plant cell? Now, both human cells and typical plant cells have 70S ribosomes. Cilia is definitely not found in typical plant cells. Plasmodesmata is actually, a typical animal cell. There is no plasmodesmata at all. It's only found in plant cells. Okay, tonoplast also only in plant cells, therefore 70S ribosomes. Which statements are correct for a typical prokaryotic cell? A typical prokaryotic cell does contain 70S ribosomes, does not contain a cellulose cell wall, contains a peptidoglycan cell wall. It contains circular DNA, that is true. It is up to 5 micrometers in diameter, also true. 1, 3, and 4, C. Diagram shows a typical animal cell. So you have a typical animal cell. You can see he's labeled the rough endoplasmic reticulum. He's saying what's the function of the membrane system labeled X? Protein synthesis, synthesizing vesicles that contain proteins, as well as I think these are the two functions. Okay, so protein synthesis and transport, definitely, because they don't just synthesize the proteins, they also package them into vesicles that are later sent through the cytoplasm, maybe to leave through the membrane. Now, what describes a lysosome? A vesicle containing enzymes enclosed by a double membrane? Totally not. Okay, lysosomes are single membraned. A vesicle containing hydrolytic enzymes and surrounded by a single membrane, found only in phagocytes. It's not just in phagocytes, it's found in in basically any cell that needs to synthesize and secrete enzymes, like maybe the cells lining the stomach walls because they secrete pepsin, or the intestines because whatever secretes enzymes. So not just in the phagocyte. A vesicle enclosed by a single membrane containing several different hydrolytic enzymes that may act inside or outside the cell. Exactly. The lysosome might act inside the cell, like in phagocytes, where the lysosome will fuse with the phagosome in order to kill the bacteria within the vesicle, or the enzyme could be secreted outside of the cell, like in the stomach. So C is your answer. A vesicle surrounded by a double membrane containing enzymes which can hydrolyze damaged organelles in a cell. Definitely not double membrane, so D is wrong. The diagram shows a triglyceride. What will be produced after the hydrolysis of this triglyceride? So if you hydrolyze a triglyceride, you're going to produce 1 glycerol, 3 fatty acids. He's saying 1 molecule of glycogen. Wrong. 1 molecule of glycerol, 2 saturated fatty acids, and 1 unsaturated. So to be unsaturated, this means that one of the bonds that you have is a double bond or more. So here we have an unsaturated fatty acid, but both of these have no double bonds. So we're looking specifically at the hydrocarbon chains. There's no double bonds here, there's no double bonds here, there's a double bond here, so this is unsaturated. These guys are actually saturated. So then we have 2 saturated and 1 unsaturated, which is B. 1 molecule of glycogen, 1 molecule of glycerol, 1 saturated, 2 unsaturated is wrong. Okay, which description lists all the components of human hemoglobin molecule? 4 polypeptides that are all the same and 1 heme group. So we said which descriptions list all the components of the human hemoglobin molecule. There are 4 peptide chains, yes, 2 alpha chains, 2 beta chains, and each chain contains a heme group. 4 polypeptides that are not all the same and 1 heme group, that's wrong. It's 4 heme groups, and yes, they are not all the same. So this is also wrong. 4 polypeptides that are not all the same and 4 heme groups, exactly D. HIV-1 protease is an enzyme that's produced by the HIV virus. two identical chains of 99 amino acids from the enzyme. In each chain, amino acids 25, 26, 27 in the sequence form part of the active site. Which orders of protein structure control the shape of the active site? So we're specifically dealing with 25, 26, 27, so definitely primary. Obviously, since here said it's an enzyme, we know the enzyme has tertiary structure. So it could be primary, secondary, tertiary. He didn't mention anything. Oh yes, he also mentioned two identical chains. So yes, also quaternary. So I would have to say a primary, secondary, tertiary, and quaternary. You can't really eliminate secondary because you don't really know if it's there or not. Which statements are correct reasons for how animals cool down in hot environments using latent heat or vaporization of water? Okay, we cool down because water has high latent heat of vaporization, which means that it requires a lot of energy to evaporate. So where is it going to get this energy from? It's going to get it from your body. So your body is really hot, it has lots of energy, it provides that energy to the water to evaporate. The water takes up a lot of energy, and so you cool down. Some animals lie down and roll in wet soil. Fish move deep in water, in deeper water. Some animals lick their fur. Exactly. So these are examples of how animals use this in order to cool down. Some animals lick their fur to make it wet because then this saliva can evaporate so that it leaves so that it cools down their body. Or some animals breathe quickly with a wet tongue hanging out their mouth. Yes, exactly. Panting. When dogs, for example, they pant, they leave their tongue out their mouth. This allows again their mouths to get wet, and so water evaporates, they get cooler. 3 and 4. But here he says it's actually one of 3 options. So obviously it's definitely not 2. Fish moving into water, this is only a trait mammals do, and they're already in water, so they should be cool enough. Some animals lie down and roll in wet soil. Exactly, they roll in wet soil, which means that they basically get themselves wet, and so this water evaporates. So it's 1, 3, and 4. C. 2 different enzymes, P and Q, are investigated to find the optimum pH for each enzyme. The results show that P works in acidic conditions, Q has an optimum pH which is slightly alkaline. Okay, so he said here that P works only in acidic conditions, and then here it's slightly alkaline for Q. So which graph shows the results for P and Q? Okay, now for pH below 7, we should find P has an optimum pH, so this should be wrong because you could see below 7 we know it's acidic, so it makes no sense for Q to have the optimum pH here. And so let's see. Okay, this is a weird looking graph, but let's see. Product concentration at 1 minute. So product concentration is high at 1 minute, which means basically that there's a high rate. But you could see again that P is at 7, and then this guy is at below 7. So this is again acidic and not the optimum pH. However, I think this is the one because Q is slightly no, actually this is 7. So this is below, everything is here below 7. So since everything is below 7, this is completely acidic. Let's see option B. Okay, again, such weird graphs, but sure. Substrate concentration at 1 minute. Now this is different because it says substrate concentration. Now substrate concentration needs to be lower, okay, it needs to decrease, and if you have a maximum decrease, this is the optimum pH, because when substrate decreases, more product is being produced, and so this is what we're looking for. So the graph, yes, it has to be upside down. Now you can see P is in acidic conditions, and Q is almost 7 and higher than 7, so it's somewhat alkaline, therefore B is your answer. Okay, 11. A student investigated the effect of substrate concentration on the rate of enzyme catalyzed reaction. The student plotted the results, so you could see rate of reaction against substrate concentration. What is the Km for the enzyme catalyzed reaction? Well, firstly, what is Km? Km is the Michaelis-Menten constant. It's supposed to be the substrate concentration at which half the maximum activity of enzyme is reached. So firstly, we need to identify the maximum activity. Maximum activity for the enzyme is where we reach a plateau against substrate concentration, and that's going to be 30. So 30 is the Vmax. We're looking for half the Vmax, which is going to obviously be half of 30, 15. So 15 is going to be someplace right here. So this is half the Vmax. Now the substrate concentration at half the Vmax, that's going to be the Km. So we go down from here, and 10, 11, 12 is the Km value. So 12 moles per decimeter cubed. There you go. Which statements could be used to describe the enzyme molecules and antibody molecules? Hydrogen bonds stabilize the structure of the protein and are important for it to function efficiently. Actually, okay, enzyme molecules and antibody molecules, hydrogen bonds could be part of tertiary and secondary structures. So this could definitely be this could definitely apply to both the enzyme and the antibody. Hydrophilic R groups point into the center of the molecule and cause it to curl into a spherical shape. This is wrong. Okay, if it's hydrophilic, it should point outwards, away from the center, towards the water, whereas the hydrophobic is going to go inside. Okay, the tertiary structure causes the protein molecule to play an important role in the functioning of protein. Of course, tertiary structure, so it's going to be 1 and 3 only, and so C. Which description identifies a reversible non-competitive enzyme inhibitor? So a non-competitive enzyme inhibitor. Non-competitive enzyme inhibitors bind to a site aside from the active site, so a different site to the active site called the allosteric site. So if this is the enzyme and it has this active site over here, the non-competitive inhibitor will bind right here to the allosteric site. So it does not attach to the active site. It can attach to a site other than the active site, exactly. It can attach to the active site and another site simultaneously, or it can attach to either the active site or another site. If it attaches to the active site, there's a big chance it's a competitive inhibitor, not a non-competitive inhibitor. So B is your answer. Before mitochondria are extracted from cells for microscopy, they are usually kept in 0.25 mole per decimeter cubed sucrose solution. He's going to say, why is a sucrose solution used? So before the mitochondria are extracted, they are usually kept in the following solution. So I have to say it's to enable the rate of reaction of mitochondria to be determined, to prevent the mitochondria from changing in dimension, to provide a source of energy, or to act as a solvent. Okay, now sucrose can't really act as a source of energy, especially if there's no enzymes available to break it down, since it can't really be pumped into the mitochondria. Okay, and that's basically not how respiration works at all, so definitely not a source of energy. Okay, to prevent the mitochondria from changing in dimension, I think is the most appropriate answer. And I feel like to arrive at this answer, you should always think and basically keep in mind that when we do microscopy, a whole lot of things are going to change in shape. If you just basically take what's called a cross section and just place it under the microscope, things are going to change in dimension. And so to prevent this change in dimension, we need to use something like a solution that is approximately the same concentration inside as outside, and usually we use sucrose for that. So don't think of sucrose as a source of energy. It's definitely not a source of energy for mitochondria. Now, the photomicrograph shows a type of blood cell, so you can see we have a red blood cell over here. He said which statements about these cells are correct. Now, some of them actually have burst around here for some reason, but the rest are perfectly normal. Now, which statements? Oxygen diffuses through the phospholipid bilayer. That is definitely true. Oxygen diffuses through the phospholipid bilayer. Sodium ions diffuse through the phospholipid bilayer. That's not true, because sodium ions are basically polar molecules, and since they're polar, they're hydrophilic, and they can't pass through the inner core, which is hydrophobic, through the inner core of the phospholipid bilayer. Okay, it needs channel proteins, so facilitated diffusion would be okay. It needs active transport, so maybe using carrier proteins, but not directly through the phospholipid bilayer. Water passes in and out of these cells by osmosis. Yes, that is true. Water does pass in and out of these cells by osmosis, so it's 1 and 3 only, which is C. Okay, which row is correct? I think I want to go back to this question for just one bit. Now you're gonna ask, okay, but why do we allow water but not sodium? Water is a much smaller molecule than the sodium ion, so it's capable of passing just fine. Moving on to 16, which row is correct for parts of the phospholipid molecule? Can be saturated or unsaturated. That's definitely the tail. The tail can have double bonds or it can have no double bonds, and depending on that, we call the tail saturated or unsaturated. Can also be found in a triglyceride. The head is definitely not found in the triglyceride. The head here, specifically for a phospholipid molecule, is a phosphate group. So since the phosphate group is not common to both a triglyceride and a phospholipid, we have to say it's the tail that's common because it's made of fatty acids. Now, a student filled dialysis tubing with a sucrose solution and knotted both ends. This forms a cylinder with a 5 centimeter and a 2 centimeter radius. Okay. What is the surface area to volume ratio? Well, let's calculate this. He said that the shape is a cylinder, and he gave you the dimension. So he gave you the length of 5, and he gave you the radius of 2 centimeters. And you should definitely have the basic formulas for a sphere, a cylinder, a cube memorized for surface area and volume. So let's go ahead and write them down. Firstly, we need to calculate surface area, then we need to calculate volume. So the surface area is going to be the area of two squares and the area of the curved region. That's 2 pi r squared plus 2 pi rl. For the volume, it's going to be pi r squared h. Now, to calculate this, 2 pi, radius of 2 squared, 2 pi 2 times 5, whereas here it's pi times 2 squared times 5. So that's going to be 8 pi plus 2 by 2 by 5, which is 20 pi. So this is 28 pi, whereas here it's going to be 4 by 5, so that's 20 pi. And now to get the surface area to volume ratio, we simply divide the two. So 28 pi over 20 pi, pi cancels out. And now if you divide them on your calculator, you get 7 over 5, which is the ratio. It's 7 to 5. So that should be somewhere here. But he says here it's in 0 points. So that's interesting. I feel like maybe they got it even smaller. So here he got them in decimals, which is definitely interesting. If you basically write this down as a decimal, it's going to be 1.4. So it's 1.4 to 1. Okay, so that's C. Now, which process does not involve mitosis? Asexual reproduction does involve mitosis. Growth of unicellular organisms does involve mitosis. Repair of tissues by cellular replacement. Now, cell replacement does involve mitosis, okay? Because you're basically replacing the cell, so there's another cell coming in its place, so it has to be by mitosis. Replacing of damaged or dead cells. This does not involve... Oh my god, okay. Growth of unicellular organisms. That does not involve mitosis. Okay, whereas the last one is basically the same as this one, replacing, so C and D are the same. The difference here is the growth of unicellular organisms. I thought it's multicellular, because if it was multicellular, then sure, yes, of course, multicellular organisms are going to divide by mitosis in order to, grow. Okay, we need more cells to grow. Whereas growth of unicellular organism is just going to involve increasing in cell size, so that's B. Okay, moving on to 19. The photomicrograph shows a cell in an onion root tip. So you have, you can see the different stages of mitosis. Now, which cells are labeled in anaphase, metaphase, and prophase? Okay, so to identify the stages, firstly, you have chromosomes just being condensed and the nucleus freshly broken down. I feel like X is prophase. Y chromosomes are along the equator, so this is definitely metaphase. Okay, and Z has the chromatids moving to opposite poles, as you can see. So that's definitely anaphase, and therefore anaphase is Z. Okay, metaphase is Y, and prophase is X. So that's gonna be D. The diagram shows an outline of mitotic cell cycles. So you have 1, 2, and 3, and then cytokinesis. So which numbered stages of the cell cycle include a period of time when each chromosome consists of sister chromatids joined by a centromere? Okay, so a period of time where each chromosome has 2 sister chromatids. Sure. So firstly, let's start by labeling. Cytokinesis is right here, which means it's basically earlier to that with mitosis. And before mitosis, we have the second growth period. Before that, we have the S phase. And before that, we have the first growth period. Now, in the first growth period, the cell has just gotten out of mitosis and it's growing and deciding whether or not it wants to move on to divide again. And so if it does move on and decides to divide, it undergoes DNA replication, which is what happens in the S phase, where instead of one chromosome with one chromatid, we have one chromosome with 2 chromatids attached by a centromere. So this is the first phase in which this happens, but it's not labeled here. You can see only 1, 2, and 3 apply to these stages. So during 1 in the first growth period, this is still true because this continues. Okay, the cell does not undo what happened in DNA replication. It continues with the pairs of sister chromatids. So yes, 1 has a pair of sister chromatids and 2 has a pair of sister chromatids. Up until obviously cytokinesis, the cell splits and there's no longer a pair of sister chromatids. So 1 and 2 only, which is A. Now the diagram shows parts of the nucleic acid molecule, and he's saying what's the name of what's the name of bond X. That's a phosphodiester bond. You can see there's a phosphate group over here, and it's joined to a carbon, and it's joined to a carbon, and so obviously an oxygen from here is going to bind to a carbon from here. So carbon oxygen bond, that's known as the ester bond, and there's two of it, and there's a phosphate group, so it's called the phosphodiester bond. That's just an extra piece of information for you, but you should know that this is called the phosphodiester bond. XNA is a laboratory-made nucleic acid. XNA is made of nucleotides in which one component has been replaced by the chemical X, and so instead of the nucleotide being, I feel like, in which one component has been replaced by chemical X. Okay, so obviously because the first component of DNA is basically the name of the sugar, so it's either deoxyribonucleic acid or ribonucleic acid. So it's either deoxyribose or ribose. Now here he's changed the nucleotide's sugar. Instead of being, for example, deoxyribose or ribose, it's something labeled X. So the chemical X is organic but not found in nature. The part of the molecule responsible for coding is not changed. So obviously the part of the chemical responsible for coding, that's going to be the bases, I feel. Yes, responsible for coding is not changed. So it still codes for the same information, so there's still the same sequence of bases. So which component of DNA or RNA nucleotide has been replaced by the chemical X? It's the 5-carbon sugar. Now, which strand of DNA does RNA polymerase bind to? So the RNA polymerase is going to transcribe the the complementary or the template strand. Okay, so that's going to be D. Okay, it's not about being lagging or leading strand, it's about which one is the template strand, and so that's going to be D. Now, which process occurs during the formation of messenger RNA? So during the formation of mRNA, we're looking at transcription. Okay, and so he's looking here at what are the processes that happen. So that's transcription. Is it replication? We're not replicating anything. Is it polymerization? Yes, we are forming a polymer, I suppose. And is it condensation? Yes, it's a condensation process. We're going to be forming dots of phosphodiester bonds by condensation, by the release of water, 1, 2, and 4b. The anticodon for the amino acid tryptophan is ACC, which shows the template DNA sequence that includes the DNA triplet for tryptophan. So to identify how this works, I think we should write down how DNA sequence is related to the anticodon. So first you have a DNA template strand, and it's going to be complementary to the transcribed mRNA. So these guys are complementary to one another. And then mRNA is going to be translated and by tRNA. So mRNA is complementary to tRNA, and so since tRNA is complementary to mRNA and mRNA is complementary to tRNA, this means that these two are practically the same, except for the fact that this is obviously RNA and this is DNA, which means that uracil is changed into a thymine and thymine is changed into uracil. Okay, and so if I have the anticodon for tryptophan, ACC, and which shows the template DNA sequence that includes the DNA triplet for tryptophan, it should be the same. Basically, it should be ACC. So if the anticodon is ACC, the template DNA should be ACC. Okay, so if you contain tryptophan, you should contain ACC, then D is the answer. The student drew a plan diagram for summative plant tissue, and the diagram is shown. It contains a mistake in the way it is drawn, so you have R, and he says the maximum width of R is 15 millimeters on the diagram. Its actual width is 250 micrometers, and the student was asked to draw a scale bar on their diagram. Okay, sure. So he said that the maximum width of R is 15 and the actual width is actually 250, and the student was asked to draw a scale bar on their diagram. Which row is correct? So the part of the plant drawn. So what do you think this part of the plant is? It's definitely not the root. Okay, in the root the xylem looks a little different. It's more towards the center. Basically the vascular bundle itself is towards the center, and the xylem looks something like this. I'm pretty sure you guys have seen it before. Whereas in the stem, yes, there is basically this okay, this is a terrible drawing, but the vascular bundles are towards the edge. Okay, so this is definitely the stem. Location of drawing mistake. So it contains a drawing mistake in the way that it was drawn, and they've labeled the mistake, I suppose, R, because the maximum width of R is 15 on the diagram, where its actual width is 250, and they were asked to, okay, so R is, I think, the one labeled, and I think it's the mistake. The maximum width of R is 15, and its actual width is 250, and I think they've drawn it to a much larger size. And so this is definitely the flume. Okay, so this part over here, this is the flume. Okay, the length of the scale bar in millimeters correctly represents 100 micrometers. The length of the scale bar in millimeters to correctly represent 100 micrometers. So to represent 100 micrometers. Okay, so he's asked to draw a scale bar because his drawing is wrong. And so what we're going to do is, if we draw the scale bar in order to make sure that this accurately represents the length that we want, the scale bar here, he said that its image length is supposed to be 100 micrometers. The actual width here was 250, and he drew in 15. So 15, convert that to micrometers, multiplied by 1,000, you get 15,000. 15,000 divided by 250, you get a magnification of times 60. So use the same magnification in this situation, and remember that magnification triangle, magnification, image length, actual length. We have the actual length, we're looking for image length. Image length equals to magnification times actual length. So magnification is 60 times 100, and that's going to be 6,000, but in millimeters that's just going to be 6. Okay, now if you're confused on what we did, we used her information because she drew wrong. She drew without writing a scale bar, and so the teacher asked her to draw a scale bar to represent this information. And so what we did is we used what she used in order to get the magnification, and then used it again here so that we can get the length of the scale bar. So this is stem and flume and 6, but we don't have that as an option. So the location of the drawing mistake, I suppose that the way they drew the xylem is wrong. Okay, like it does not have these striations. Okay, it's just yes, a little darker, it has cells, but it's definitely not striated like this. I thought that R was just labeled because it's the mistake, but I feel like xylem is the one that's drawn wrongly. Okay, you're not supposed to shade in your drawings. So stem, xylem, and 6. I know this question is a bit confusing, so make sure to take your time while solving it. 4 students have drawn and labeled a large part of the structure seen on electron micrograph. Which students have labeled their drawing correctly? A. Mitochondria, sieve plate, thin cytoplasm. That's definitely a correct labeling. Okay, mitochondria, this is the sieve plate, thin cytoplasm. A is correct. Let's see the other options. B. Vacuole is definitely wrong. The nucleus, I feel like the nucleus is definitely bigger in size. And chloroplasts, sieve plates do not have chloroplasts, and so it's A. Okay, the diagram shows some structures here used to transport the flume. Cell Y, companion cell, direction of transport goes from here to there, and this is cell Z. So this will be the source, this will be the sink, and then he said here which statement is correct. Cell Y is the sink cell. Cell Y is the source because the direction of transport is this direction, so no. Cell Z can be above or below cell Y. Cell Z, cell Y, yes, sure. Translocation happens in both directions, up and down, so it doesn't really matter. Cell Z must be a photosynthetic tissue. Cell Z does not need to be a photosynthetic tissue because it's the sink, it's the receiving end. Okay, cell Y allows glucose to move to the sieve plate, the sieve tube, via the companion cell. Allows glucose to move to the sieve tube via the companion cell. I suppose, yes, glucose could move from the companion cell to the sieve tube, but what's mostly being translocated is actually sucrose, not glucose. So I think that the most correct answer is B. And cell Y allows glucose to move to the sieve tube via the companion cell. It's I feel like it's just a little too simplified for my liking. Okay, moving on. The diagram shows a section through a root of a dicotyledonous plant. So you have a xylem, Casparian strip, root hair cell, and soil particle. Now, which statement describes the movement of water through and solids through this through this pathway? So you have a soil particle and you have water moving from the soil into the root hair cell via osmosis and ions via active transport and everything. They continue moving across, I suppose, the cell wall, the Laplace pathway, until if some water molecules are just moving like this, they face the Casparian strip, they can't continue to move, so they have to move through the cytoplasm and everything. And because most water prefers to move in here, it prefers to move in the cell wall, since the cell wall is part of the apoplast pathway, the dead pathway, which has less resistance compared to the cytoplasm and the what you call cytoplasm and tonoplast and everything, since they obviously contain a whole lot of materials and particles in them. And so here, which statement describes the movement of water and solids through this route? A layer of suberin causes water and solids to move from the uplast pathway into the symplast pathway. Exactly. Okay, a layer of suberin causes water molecules to move from the symplast to the uplast. That's wrong. Okay, they were already moving in the uplast and then they move into the symplast. They were moving in the cell wall, but then suberin is a waterproof material. It forces the water to leave the cell wall and move into the cytoplasm, which is the symplast pathway. The tonoplast causes water and solids to move. I suppose this is the same drill, so A is your answer. Okay, the graph shows the effect of different partial pressures of carbon dioxide on the oxygen dissociation curve for hemoglobin. So you could see when the partial pressure was 1, the curve was a little higher up. When the partial pressure was 1.5, the curve moves down and towards the right. So he says here, what is the change in percentage of oxygen saturation of hemoglobin at partial pressure of oxygen 6 kilopascal as the partial pressure of oxygen changes from 1 to 1.5? So it's just a calculation question. Firstly, let's go to 6 kilopascal. So at partial pressure of 6 kilopascal, at partial pressure of CO2 of 1, we had a 70% saturation, whereas for this graph here with partial percentage of pressure of 1.5, it was 50%. So the change in percent was 70 to 50, so it was 50 minus 70, which is a decrease by 20%, exactly. Now, what happens in the heart of at the start of ventricular diastole? So at the start of ventricular diastole, what happens is that all the valves are open, obviously except for the semilunar valves. Okay, the valves are open, so the anteroventricular valves are open, and the blood just fills in. Okay, so atrioventricular valves are open, that is true. Semilunar valves are open, that's wrong. The pressure in the atria rises above the pressure in the ventricles. No, that happens during atrial systole, which is a different phase. Okay, ventricular systole, diastole is part of generalized diastole, and so here we don't have any rising in pressure. The pressure is just dropping. The pressure in the left atrium rises more, no and no, so it's B. Okay, the electron micrograph shows a longitudinal section through the blood vessel. So you have a blood vessel, and he's saying here that the electron micrograph shows a longitudinal section. So which type of blood vessel is shown? Now, I suppose since we can see individual cells and not many layers, just like a single cell and only red blood cells passing through, this is definitely a capillary. Okay, B. A student drew a sketch to show the formation of tissue fluid. Which label is correct? Blood enters capillary and pressure increases. Red blood cells and water move into tissues. Higher water potential, so water moves back into the capillary. Higher volume of blood returns to vein. Okay, firstly, red blood cells do not leave the capillary. There's red blood cells here, red blood cells stay. So B is definitely wrong. Okay, blood enters capillary and pressure increases. Now, I would guess that yes, the pressure is high at the arterial end, but does the pressure change? I mean, it's already high. It's not like get higher. Like, pressure at this end is constant, pressure at this end is constant, and pressure in the middle is constant. It's high here, low here, and middle here. Okay, now higher water potential in tissue fluid, so water moves back into the capillary. I suppose that's right, because here water is going to move into the tissue fluid, and then water potential and basically lots of solids accumulate here, and so stuff moves back there because things like carbon dioxide, these things, they're going to leave, and so will the water that's just entered, because there's a high hydrostatic pressure on this end and low one here, so C is true. Oh my god, what did I do here? Okay, lastly, we have also a higher volume of blood returns. Again, it is the same volume of blood that returns to the vein, because overall things leave and things go back in. Structures are present in the trachea and also in all bronchioles: cartilage, smooth muscle, epithelial cells. Okay, in trachea we do have cartilage, we have smooth muscle, and we have epithelial cells. In bronchioles we don't have cartilage, but we do have smooth muscle and epithelial cells, so it's 2 and 3 only. The photomicrograph shows a section through part of the gas exchange system. Now, which tissue can be seen? So you can see a cross section of I believe these are these are some cells, definitely. Okay, we have a cross section, and he says here, is it what tissue can be seen? Can you see cartilage? Can you see ciliated epithelium? Can you see smooth muscles? Can you see squamous? So I feel like this is the alveoli. So this is the cross section of like the lungs and like the alveoli. And so what happened, what I believe we're seeing here is just squamous epithelial cells. We don't see any ciliated epithelium. If we see ciliated epithelium, we'd see the cilia. If this was cartilage, you would see really thick layers containing these chondrocytes. If it was smooth muscle, you would see the smooth muscle. So what's the function of goblet cells in the gas exchange system? To produce mucus, okay? So to release mucus. Which organisms have cell walls that can be affected by penicillin? Protoctis, not bacteria, not viruses. Firstly, bacteria have peptidoglycan cell walls, and penicillin only affects peptidoglycan cell walls. Protoctists have cells made of, I believe protoctists, I think that depends, but yes, they're not made of peptidoglycan. And viruses definitely do not have any cell walls, so it's just one. Okay, each year there are 462,000 deaths from malaria in children under 5 years old globally. Insecticide-treated nets could prevent 50% of malaria cases and reduce deaths of children under 18%. How many children could be saved using insecticide-treated nets? Okay, so insecticide treatments could prevent 50% of malaria cases and reduce the deaths of children under 5 years old by 18%. I'm saying how many children could be saved using malaria. So if we are preventing 50 cases of malaria and reducing children's deaths, now we're specifically talking about saving children from, I suppose, death. So we're going to use the 18%. Okay, so 18 times 46200 divided by 100 is going to be 83,160, which is B. Which row is correct? Involved in phagocytosis secrete antibodies. Okay. So involved in phagocytosis are they T lymphocytes or B lymphocytes? Now T lymphocytes are are involved in phagocytosis. Secrete antibodies are going to be plasma cells produced by B lymphocytes. So A is correct. Now 4 steps that occur in the primary immune response to a pathogen are listed. P, B lymphocytes develop mitosis. T helper cells interact with macrophages that help digest pathogens. Plasma cells secrete antibodies. T helper cells activate the other lymphocytes. These steps can be arranged in the sequence in which they occur. What will be the third step? Okay, first things first. Antigen presentation. Yes, macrophages are going to basically digest pathogens, get once the pathogens enter, the macrophages are going to present some of the antigens of the pathogen. Now once that happens, T helper cells recognize these antigens, so this is a T helper. So when they recognize these antigens, what happens is that they are going to be stimulated, and this results in their clonal selection, and then this is followed by them dividing, clonal expansion, and then they start to alert everyone else. So Q is step number 1. Step number 2, T helper cells will activate the other lymphocytes. Exactly. T helper cells are then they're going to go around and stimulate other lymphocytes, that's S, and so B lymphocytes will be stimulated to divide by mitosis and to produce plasma cells. Plasma cells then secrete antibodies. So P then R. So Q, S, P, R, which is, what is the third step? It's P. A. And this is it for this video, guys. I hope this video was useful to you. If it was, make sure to like, share, and subscribe. And I hope to see you guys in my next one.